Hi, everybody. I'd like to introduce our first speaker in the before lunch part. It's me. Hi, I'm David Tabb. I am obviously an American by the weirdo accent, but I moved here 2 years ago to join the Division of Molecular Biology and Human Genetics over at Stellenbosch University. And because the Medical Research Council is paying a lot of the bills, I actually shuttle around Cape Town a fair bit. So on Tuesdays, you can find me over at the med school for UCT, and on Thursdays, you can find me over at the University of the Western Cape. It's been a lot of fun because it's an exposure to a lot more students and a lot more kinds of projects as well. But the place where I've spent the last 20 years of my career is proteomics. And I thought that it would be really valuable to spend a little bit of time with the subject of why people should bother with proteomics. You might think that proteomics is kind of a new and amazing technology for South Africa, but in fact, we have a pretty long history in this area. And in 2008, there were already 3 laboratories in country doing this, and now we've counted 18 different laboratories across South Africa that are capable of doing either metabolomics or proteomics. So I think it's worth talking about what these facilities can do for you and what you have, what rationale might really require that you add this into your armamentum for handling molecular biology. So I've tried to answer the question of my title 4 different ways, and I'm going to walk into these as we go through the talk, and I hope that that's going to be useful for you all. So we start with this idea that proteomics is closer to the coalface. Now, somebody who knew as much about mining as I did when I came to this country might have no idea where that expression comes from. But a coalface reflects the fact that in a big coal mine, not all of it is actually operational. You've got a huge amount of a coal mine that's just room for people to get things from point A to point B. The coalface represents where the work is actually getting done, where coal is being chipped from the active face, from that seam in the earth, and where the material is coming out. So when you study proteomics, you're getting much, much closer to where the work is actually happening on the metabolic landscape. Because every reaction that has its friendly enzyme there to catalyze that reaction is part of the action on the cellular stage. And if you want to measure that directly, at least from things that have sequences, proteomics is as close as you're going to get. Obviously, metabolomics is yet another stage, but now we're not looking at things with sequences. It's a much harder place to work. So if you want to be where the action is on the molecular stage, studying proteomics makes all kinds of sense. Many, many biological processes are heavily dependent on post-translational modifications. What are you going to learn about post-translational modifications from a sequencer experiment? Nada, or very close to nada. Some of them are sequence dependent, of course. So this is one of these things where PTMs are obviously very important for us to understand if we want to know about the action at the cellular stage. Plenty of time, I see people working in non-model organisms. Now, with our wildlife TB group within our division, we see an awful lot of people who are working on species for which you can't buy a kit. We're going to talk about an example of that. And finally, if you're involved in biomarker research, one of the things that we like to be able to do is to translate all this molecular fishing that we do into something that we can use clinically. And it's really hard to beat ELISA for that. So I'm going to talk a little bit about some of the challenges we've seen there. All right, now everyone's seen the central dogma here. So think about where these measurement technologies fit in the context of the central dogma. If you're doing DNA sequencing, yes, you can find all kinds of interesting variants, some of which are coding. And if you're doing that, you may be able to find some mutation that is relevant to a biological process. That's grand. But how readily do you think, how easily will you translate that sequence variance into something like function? It's actually a lot harder. To say that something is correlated with function is nice, but wouldn't you rather have a biological mechanism that ties it all together? If you look at tools that we have for evaluating whether this particular sequence variant has any impact on function or not, you're going to find there's an awful lot of discrepancy. you could be running SIFT or you could be running polyphen on exactly the same set of mutations and come up with a completely different assessment of which of those actually have some bearing on activity. So that's valuable to be thinking about. Next step, RNA-seq. How do the genes being transcribed differentiate this tissue? So this is very powerful because, of course, which products from our molecular catalogs get ordered in a particular tissue is going to differ an awful lot. So by moving from DNA sequencing to messenger RNA sequencing, we're getting a lot more information about this process of expression. What is it about this cell type? What is it about this tissue that is allowing for this type of behavior? And superficially, protein identification by tandem mass spectrometry is a very related technology in that it's giving us a kind of an inventory of all of these different proteins that might be manufactured by a tissue or by a cell type or whatever. So you might ask, can you do single-cell proteomics? And I would say that the answer is pretty authoritatively no so far, although I'm sure that there are some very large hat proteomicists out there who'd like to tell us that they can. But we can work from vanishingly small samples at this point. And the example I would use for this is laser capture microdissection. I think that one of our speakers mentioned that just a bit this morning. And sure enough, yes, you can work with a few caps of protein, of cells that have been abstracted out of a more complex tissue by laser capture microdissection. So even very small amounts of samples can be translated into meaningful protein lists. So how do we do that? Now, I'm a bioinformaticist of biological mass spectrometry, not so much the person who runs the instrument, but I hope that this can help sort of spill out all the different processes that are required for these. One of the first things that people dislike about proteomics is that we start by cutting them all up. We might start with a bunch of intact proteins, and intact proteins are what we want to get back out, but in fact, intact proteins are a pain to deal with, both in terms of separation and in terms of their behavior in a mass spectrometer. So one of the first things that we typically do is digest up these mixtures of proteins into mixtures of peptides. Frequently, we have tens of thousands or hundreds of thousands of different peptides that we need to differentiate from each other. And something like that is too complex for us to deal with in a single LC-MSMS experiment. This is the term that gets thrown around. That means liquid chromatography tandem mass spectrometry. And I've got all those elements drawn out here, but we'll get to those. So one of the first things that you frequently see is that people will try to fractionate these collections of millions or hundreds of thousands of different peptides into different fractions on the basis of some property. One of the old school ways to do it was on the basis of strong cation exchange or maybe Hillic. We have all kinds of different means for this purpose now. So if you can separate your original samples into, say, 24 fractions or 96 fractions if you're being very extreme, you can then subject each of those to LC-MSMS, which is what the rest of all these little icons represent. The instrument is a serial technology. It's a serial sampling technology. We're going to repeatedly look for the ion that hasn't been explained so far that's got the most intense signal. This represents a particular peptide coming into the mass spec. And we're going to subject it to tandem mass spectrometry, isolate a whole bunch of copies of some peptide, beat the heck out of it by making it move very quickly in the presence of gas, and then look at the fragments that come from it. That's what all these other icons represent. So we separate them in time on the basis of hydrophobicity. We electrospray them. This is a little like having a cattle prod tied to the end of a water hose. That creates a plume of ions. We then measure the ions two different ways. A very accurate mass measurement of the intact peptide. We blow up, we isolate a bunch of ions of that peptide, beat them up by bouncing them off of gas molecules, and then we collect the list of fragment ions that they produce in a tandem mass spectrometer. That is the input set for the bioinformatics that give us back our protein lists. And for once, that's not the lecture I'm giving. So we're going to just remember that this is the basic strategy that most people are talking about when they say proteomics. More specifically, this is called shotgun proteomics, or proteomic inventories. So you might ask, why is that necessary? If I can measure the amount of messenger RNA in the cell, don't I know how much of the corresponding protein is there? I wanted to assemble one slide that gave some of the most prominent reasons why a measurement of messenger RNA does not tell you that much about the quantity of the protein present in the cell. So we have synthetic regulation on the left side and catabolic regulation. You can think of this as an input and an output. What are the things required to get from a messenger RNA to a mature protein, and what are the processes that are getting rid of those proteins on the other end? So first off, messenger RNA in its own right has a complex structure, especially once you've got things like RNAi and so on interfering with that process. So you need a messenger RNA that actually associates properly with a ribosome. It might be that a particular messenger RNA has a whole bunch of oddball codons in it, codons that are not the most frequently found version of that tRNA for that particular amino acid. So if you have codon bias, your pool of transfer RNAs is going to slow down the process of synthesizing a protein from it. Certainly, folding a polypeptide is a very big deal. Just having a mature polypeptide that's flopped off of the ribosome does not mean that that protein is now active. So taking on a structure, whether that's an assisted process or not, matters. Certainly, a lot of proteins have sort of a shipping label at their N-terminus. And if that shipping label is still intact, that tells you that this protein has not been transported to the proper place where it should be in the cell. So cleaning off these propeptides is one of these markers we have that this shipping and handling process is over, and the transporting there. And of course, modifying it for activity. Lots of proteins are completely inert until we've got them modified in such a way that they can swing into action. So at the other end of that, we have a lot of different processes that lead to proteins being taken out of the pool. Certainly, if you spend any amount of time studying lens crystalline proteins, you've seen some proteins that have seen it all, quite literally. Well, they've been exposed to air over years of time, and you end up with all kinds of crud hanging off of these proteins, some of which get them marked for recycling. It may be that something that takes place that denatures the structure of the protein. Now, if you're simply measuring the peptides that are seen for that protein, you're not going to have a huge amount of information about the structure. But it's still part of this sphere of things that causes a protein to be useful or not. Certainly, our cells have processes for marking proteins that are good to be put out to pasture. And ubiquitin is one of these really common mechanisms that a lot of people study now. You can evaluate from proteomics data which proteins have been marked for destruction by looking for these ubiquitin tags on them. So these are things that you are never going to see if you're only studying by messenger RNA, that you can with proteomics. Finally, there are lots of ways that our cells get rid of proteins that shouldn't be in operation anymore. And I've shown a very large structure here at the bottom right to remind us of the proteasome. This is in itself a protein complex that takes as its input intact proteins and produces basically garbage amino acids at the end. So this entire process can be thought of as turnover. You have an input for new proteins being synthesized, and you have an output of proteins being shuffled off to greener pastures. It is possible to go quantitative with proteomics. And one of the things I have to caution people about who are taking on proteomics for the first time is that if you want a strongly quantitative result, you should do a quantitative experiment. Now, 2 slides ago, I showed the most common workflow, the data-dependent shotgun paradigm. But we have a lot of methods within proteomics that are quite a lot better at giving you strong measurements of quantity for these proteins that don't bounce around a lot. So if you want a coefficient of variance, a CV value of 10% or less, you'd probably better focus on getting very strong measurements of your particular proteins of interest and not broadening your scope to try to measure every protein in a cell simultaneously. That does not lend itself to very invariant measurements of quantity. So in SRM, we again start with a peptide mixture. We've digested the proteins up. We're still using liquid chromatography and electrospray. But now we're configuring the instrument quite differently, and often we're using an altogether different cohort of instruments for this purpose. I believe earlier you may have mentioned a triple quadrupole instrument. So a triple quadrupole is a type of mass analyzer that we use for doing quantitation experiments, something called selected reaction monitoring. You'll often see it called multiple reaction monitoring, but honestly there's no difference between the two. So we're going to set quadrupole 1, the first part of the instrument that the ions are flying into, to screen out everything but the intact mass of the peptide. In the second quadrupole, we're again conducting this process of collision-induced dissociation. We're bouncing the peptides off of gas molecules until they break apart. And then in the third quadrupole, we specify that we only want to see fragments of a known mass. So obviously you're going to have to know what is the peptide that you want to measure and what fragments dominate when you break apart that peptide ion. So this Q1, Q2, Q3 process helps you to create what's called a chromatogram, which is a signal that rises and falls as that peptide first appears in chromatography, comes to a peak, and then falls off with time. Often that window is only 30 seconds out of a 90-minute experiment. So you might have 50 different proteins for which you've decided to measure peptides, and from those you're going to get very accurate quantitations that say how much of this peptide is present in this mixture. Ideally, you'll be measuring multiple peptides per protein as well. And perhaps it's obvious to everyone, but post-translational modifications are quite essential to biological functions. So I've drawn over here just an EGFR receptor, and I want to remind you that at the start of this signal transduction process, all of the proteins are present, but they're sitting around waiting for a signal. Now, if you have a signal that arrives, you need to be able to do something with that signal right away. And so having these proteins manufactured and ready to swing into action matters a lot. You can't wait on a whole new gene expression to produce a whole crop of new proteins before you handle the signal that's arrived at this particular moment in time. So this is largely driven by phosphorylation events. And phosphorylation gets a very disproportionate amount of attention in the proteomics community. People have invested tremendous time in making the best possible inventories of kinases, of all of their targets, of what kinds of, how prominent is each type of phosphorylation. It's an amazing field. And yet, there are other types of post-translational modifications that also come into play. I mentioned a while ago that you can do proteomics in the aftermath of formalin-fixed, paraffin-embedded samples, which means that hospitals who have these huge vaults filled with FFPE samples could be using those as inputs to proteomics. But formalin fixation and paraffin embedding does a lot of interesting things to the underlying peptides. I already mentioned that the lysine peptides tend to be harder to spot because they've been disproportionately affected by the cross-linking. So if you want to look at these things, one of the things you'll need to do is to screen out all of the post-translational noise, the modification noise that the FFPE reaction itself entails. Okay, so in the case of lots and lots of biological processes, we see that the changes that are mediated through these very fast responses come through post-translational modification, not through fresh expression. So why is it that a post-translational modification can be visible to us through tandem mass spectrometry? So we start with we'll just use the very simple example of a phosphorylation. Now I've grabbed a peptide here from alpha casein. It's just a protein that carries a lot of phosphorylation on it. And we see that in this case, our short peptide has 3 different places where that phosphorylation could attach. We've got a serine in position 4, a serine in position 6, and a threonine in position 7. So is it enough to know that we have this peptide bearing 1 phosphorylation or 2 phosphorylations? Typically not. Typically, when people are studying phosphorylation, they care very much which explicitly of the amino acids in the sequence are bearing the modification. So I want to talk about how that impacts the data produced from such a peptide. So we start with the fact that each phosphorylation is going to add 80 Daltons to the mass of the peptide. So can we tell apart the modified and the unmodified peptide by high-resolution mass spectrometry? Yes, absolutely. Our instruments are more than capable of discerning a single neutron difference in a peptide. So something like 80 Daltons is going to be very far apart. Those are completely different worlds to a mass spectrometer. Next, we observe that fragments that contain the phosphorylation are going to be higher by 80 Daltons. So let's try to walk through what that means. In this process of collision-induced dissociation, we break apart the peptides, but ideally, we're breaking each copy of the peptide in just one place. So if we imagine this top peptide, the one without any modifications, it might break at the 4th peptide bond. So 1, 2, 3, 4. So we would have, 1, 2, 3, oh, yeah, okay. So we have the DIGS, which is the B4 ion. It contains the left end of the peptide, and it contains 4 amino acids, thus B4. And then we have the rest of it, which becomes, in this case, a Y5 ion. It's the C terminus of the peptide, and it contains 5 amino acids. So when we look at what this looks like, if we wanted to discern whether the first serine or the second serine were bearing that phosphorylation, we would ask, does the B4 ion contain the mass of phosphorylation, the phosphorylation, or does it not? Does the B5 ion contain the phosphorylation, or does it not? And does the B6 ion contain the phosphorylation? And you can also do this from the other end of it. So we're not just seeing a shift in the mass of the whole peptide, we're also seeing a shift in the fragments that are produced from these. Of these, we see that one peptide can become 4 different intact masses, because you can have 0, 1, 2, or 3 phosphorylations sitting on board this thing. And within the set that would have the same peptide mass, you can differentiate them on the basis of their fragment masses. Okay, so that is a useful examination. So I wanted to point out a couple of ways that this can become useful. So we have a wildlife TB group that is doing some fascinating research in all kinds of different animals that we have here that do or do not suffer from tuberculosis in the wild. Not MTB, I guess, in this case, but is this M. bovis? M. bovis, okay. So if we have a black rhino, we might want to evaluate whether the black rhinos are having an immune response that is suggestive of a TB infection. So one of the things that we might want to do then would be to measure cytokines and chemokines by some kit. So QIAGEN has a qPCR kit that we can use, but it's designed for a horse. So why might we go ahead and buy the QIAGEN kit? Because we haven't got anything else. We haven't got anything else. If all you know how to measure is horse cytokines, and a horse is the closest organism to the one that in the wild that we want to measure, then we bloody well might use the horse kit. Well, that's not ideal in a lot of reasons. So having looked at some of the data from the public assembly of the white rhino, I see that some of the sequences that this kit should be measuring are actually quite different than what they should be for horse. So if you're making a measurement with this kit and you want to know which parts of it are or are not working for this species, you better start from the sequence perspective. Now, one of the things that I wanted to note about the selected reaction monitoring experiments, the quantitative mass spectrometry experiment I described just a moment ago, is that they're customizable. If you know the sequence of the thing, you can change the fragment ion masses that you're looking for. So if you can see it in a mass spec, you can measure it. And there's no requirement that you customize, that you buy some product specific to each species in order to do the experiment. So ideally, if we can move our measurements of cytokines and chemokines to a selected reaction monitoring platform, we will no longer have the requirement that we've got to order an expensive kit from a vendor for the inappropriate species. Okay, so on we go with the next set. One of the things that is that is a bit of a surprise to some people is that I've only been working in tuberculosis for 2 years. The 10 years before that, I spent in the world of colon cancer out at Vanderbilt University. And one of the things that we did was to be part of the Early Detection Research Network. Being part of EDRN meant that every half year or so, we would show up at these meetings and see talk after talk with ROC curves, showing how great a particular biomarker combination was for detecting an early stage of cancer, for prostate cancer, for pancreatic cancer, for ovarian cancer, whatever, just all these different cancers. Look at all these great curves. We've got biomarkers for everything. In the end, lots of these studies failed. When it came to the next stage, how can we verify that this set of biomarkers is viable in a different population than the one on which it was trained, right? So I want to point out that one of our goals here is that we get to a clinical test. How likely do you think it is that we will draw some blood from our patients in the field and run it through a mass spectrometer to figure out whether or not that protein is present or not? Not bloody likely at all. The reason is that mass spectrometry is a complex bit of equipment that needs to keep its insides at a vacuum. They're big, they're expensive, they require hard-to-train operators, and so on. It's problematic. But we have better alternatives for measuring proteins in the field. And ELISA, these sandwich assays that we use for immune measurement of particular targets, are a great option. So if you have an assay that you can do by selected reaction monitoring, and you want to translate it into a test you can use for actual patients in the field, this selected reaction, well, think of it as the discovery proteome giving you candidates that might be differential, selected reaction monitoring that's telling you that, yes, these particular biomarkers are a very good set for clinical decision making. You then create an ELISA assay based on that SRM platform. Now you have an assay you can use in the field. So this is the kind of biomarker pipelining that we need to be thinking about. If you have a brilliant signature for microRNA that is perfectly good at differentiating one population from another population, that's wonderful. But in fact, that's going to be quite difficult to turn into an assay that you can use every day on patients. So proteomics has this strong link to the downstream clinical testing field. And we hope to see that this transition is one that more and more teams are making successfully. So if you want direct evidence of catalysis, it's awfully nice to be able to measure that the enzyme itself is present, not on the basis of messenger RNA, but on the basis of a protein expression. Proteomic inventories seem to get all of the attention in our field. A lot of people have invested years and years of effort in identifying more proteins. This has been a mantra for us for 2 decades now. That is starting to change. People are starting to think of proteomics as a quantitative exercise, and there's more and more effort being placed into this space. Post-translational modifications are invisible to sequencers, but if you have a proteomics facility nearby, you can be measuring those as well, because of the differences not just in peptide mass, but also in the peptide fragment masses. Proteomics methods are adaptable to your species, so long as you have a genome assembled. If you don't know the set of proteins that exist for a particular species, proteomics is a lot harder. But we're now starting to do some work over at University of the Western Cape on the identification of proteins in species for which we don't have an assembled proteome. And I'm kind of excited about how that's turning out. So with that, have I run over time? Oh, great. Do we have a question from the audience?